Dear students, we have to pass on to the next part of classical genetics, that is cytoplasmic inheritance. It's also called by name extrachromosomal inheritance or extranuclear inheritance, all same. Cytoplasmic inheritance, extrachromosomal inheritance, and also extranuclear inheritance. As you know, the DNA is a genetic material, universal genetic material. See, we have the DNA in the chromosomes, and those genes located in the nuclear chromosomes, the meaning chromosomes found in the nucleus, follow the Mendelian inheritance. They are inherited through the genes present in the chromosome, that is called nuclear genes present in the chromosome. So, the nuclear genes located in the chromosomes follow the inheritance, what's called Mendelian inheritance. But there are certain traits whose actually character are governed by genes located either in the chloroplast or in mitochondria. Those organelles present in cytoplasm. So, there are certain traits whose genes are located either in the chloroplast or mitochondria, controlling the characters. This is called cytoplasmic inheritance. Otherwise called extrachromosomal inheritance. Why is it called cytoplasmic inheritance? Now the chloroplast and the mitochondria are the cell organelles present in the cytoplasm. They carry the genes for the expression of characters, hence called cytoplasmic inheritance, as these organelles are present in the cytoplasm. Either the chloroplast or mitochondria, they have their own DNA, circular DNA. They can express some of the characters. That's why these organelles are called semi-autonomous organelles. Semi-autonomous organelles. As they have their own genes controlling the expression of certain traits, hence called semi-autonomous organelles. In both the cases, we have circular DNA. Now, this is due to the presence of certain units in the chloroplast and mitochondria. These units are called plasmogenes. So these plasmogenes are present in the cytoplasmic organelles like chloroplast and that is mitochondria, and these are responsible for the expression of the characters. So it is called cytoplasmic inheritance because the inheritance takes place through the cytoplasmic organelles, not through the chromosomes present in the nucleus. Now this is one. Now we have to go for first one, the chloroplast inheritance. It was studied in 4-o'clock plant, what we have the example for incomplete dominance. So concurrence studied the chloroplast inheritance. So we have to take the chloroplast inheritance. Number one, chloroplast inheritance. Chloroplast inheritance. In 4-o'clock plant. You know that one, Mirabilis jalapa. Now, in these plants, he observed two different types of variegated leaves. So you know the meaning of variegated leaves. A leaf having patches of green and then white. But when you are talking about variegated leaves, the meaning for that one, different types of leaves. So one leaf is having dark green, that is leaf. So a plant with dark green leaf. Another one, that is a plant with pale green leaf. Pale green leaf. Pale green leaf. Now, he did reciprocal cross as did by Mendel. So in the case of reciprocal cross of Mendel, in both the processes, we have received the same phenotype. But what would happen in the case of reciprocal cross between a dark green leaf and pale green leaf? In 4 o'clock plant, 2 types of variegated leaves are seen. One plant with the dark green leaves, another plant with what is called the pale green leaves. So we can have dark green leaved plant and pale green leaved plant. In the first cross, what he did, he transferred the pollen grains from the dark green leaved plant to the stigma of pale green leaved plant. So this is a female plant and this is a male plant because the pollen grains are transferred from here to the stigma of this plant. What would happen? So if it is the dark green a dominant character as per the Mendelian principle, we are getting what is called dark green. Now in the F1 generation, here what we obtain, pale green leaved plant exhibiting the character of the female plant. Exhibiting the character of the female plant. Now a second cross was effected where the pollen grains from the pale green leaved plant are transferred to the stigma of dark green leaved plant. Here the dark green leaved plant is acting as the female plant. Now this pale green the male plant. What is the result? So if it is a Mendelian reciprocal cross, in both the processes we have to obtain only one variety. All the plants should be of same type or identical. But here, in the case of reciprocal cross, the F1 plants appeared differ from each other. In one case, we are getting what's called pale green leaved plant. In another case, we have received dark green leaved plant. So in this cross, you can find out the appearance of one character. Only one character is revealed. Only one character is revealed, and that too, the character from, that is the female parent. So here it is not because of the genes present in the chromosomes, what we can say nuclear genes. So in both the crosses, you see that one, there is a difference in reciprocal cross. Difference in reciprocal cross. So in the case of Mendelian principle, if you are doing the reciprocal cross, when the sex is reversed, you can get only one type. But here, when you are doing what is called the reciprocal crosses, in both the ways, when sex is reversed, in one case we are getting pale green leafed plants where the female parent is pale green. In another case, we are getting what's called dark green leafed plant where the female parent is dark green. So in this cross, the character which appeared is nothing but the character that is used by the female plant. So the character from the female plant is transferred. This is because of not the nuclear genes. If it is a nuclear gene present in the chromosomes, we are getting just the same result in both the processes. But here, it is due to the genes present in the chloroplast. So a gene present in the chloroplast, from the female plant, the ovum with this gene is actually involved by providing the cytoplasm, whereas the male is providing only the nucleus and no cytoplasm. As the female parent provided the cytoplasm, along with the cytoplasm we have the chloroplast also transferred, and that contains the gene. That gene is responsible for the expression of the character. As we have only the ovum of the female plant provided the cytoplasm, the character is decided by only the female. So it is called maternal inheritance. So cytoplasmic inheritance or extrachromosomal inheritance or extranuclear inheritance or maternal inheritance, because only the female plant provided the character for reappearance. Because during fertilization what will happen? The sperm provided only the nucleus, whereas the egg provided the nucleus along with the cytoplasm. That's why the organelles also transferred to the zygote during fertilization and expressed in the F1 generation. So it is because of cytoplasmic inheritance, not because of nuclear inheritance, as in the case of Mendelian principle. Here, the character is transferred from the female plant to what is called the F1 generation. Here also, the character is transferred from the female plant to the F1 generation. That means the female plant provided the ovum, which contained the cytoplasm with organelles, which are being transferred to the zygote during fertilization process. So anyway, the character is decided by the cytoplasm, the cytoplasmic genes present in the mitochondria or in the chloroplast, and not by the nuclear genes present in chromosomes. Before to study the cytoplasmic inheritance, that is mitochondrial inheritance in maize plant, we have to go for the cellular organization with reference to 4 o'clock plant. When the cells are organized, when the cells are normally just actually analyzed, it shows the different types of actually cells. Now it is a variegated leaf having what we have the pale green region and dark green region. Now in the dark green region we have the cells, these are all the cells having chloroplast. In the case of dark green region, we have in some cells both green chloroplast and white chloroplast are present. In the case of dark region, when the cells are analyzed, the cells show both, that is green chloroplast as well as white chloroplast. Or the cells also reveal presence of only the green chloroplast. So anyway, now in the region, the dark green region, we have the cells having both the type of chloroplast or only one type of chloroplast. If you are taking another region, what is called the pale green region, the cells show only what we have white chloroplast. That is why that region is pale green in color. This is a cellular organization about the 4 o'clock plant regarding, that is cytoplasmic inheritance, the chloroplast inheritance. What is the reason for the development of variegated leaf? So the green region has chloroplast of both the types in the cells or only one type, that's a green chloroplast. But in the case of pale green region, we have only the white chloroplast, that's why that region is pale green in color. So this is with reference to 4 o'clock plant. Male sterility in the case of pale maize, so this is a pale maize, the binomial sorghum vulgare is the best example for mitochondrial inheritance. So here actually the female plant produces sterility in the case of male. That's why it's called a cytoplasmic male sterility caused by the gene present in the mitochondria of female. So this male sterility is normally inherited maternally and the genes are present in the mitochondria. So here the cytoplasmic inheritance is studied by using the male sterility in the case of pure maize, that is sorghum vulgare. And there it is an example for cytoplasmic male sterility. Here the male sterility is inherited maternally, that is the gene present in the female plant is responsible for male sterility. That gene is located in the mitochondria, hence it is an example for cytoplasmic inheritance. If you are taking the maize plant, there are two types of cytoplasm. Types of cytoplasm. There are two types of cytoplasm. Number one, normal cytoplasm. Normal cytoplasm. This is represented as N. Another one, aberrant cytoplasm. Aberrant cytoplasm. This is designated as S because it results in sterility in the case of male. These are the two types of cytoplasm. Suppose a cross is effected between normal cytoplasm of male and the aberrant cytoplasm of female. What would be the result? Likewise, that is, we are taking the female plant, that is normal cytoplasm, and aberrant cytoplasm in the case of male plant. That is, we are doing the reciprocal cross as in the case of Mirabilis gelata. Here too we have found, that is, reciprocal differences between the individuals in F1 generation as in the case of Mirabilis gelata. Now what is a cross effect? Let us take two different types of crosses, the reciprocal crosses. One, see a male with a normal cytoplasm and female with aberrant cytoplasm. Second, a male with aberrant cytoplasm and a female with normal cytoplasm. These are the two reciprocal crosses. What is the result we are getting? Let's see the illustration. Male sterility in maize plant, that is sorghum vulgar, is the best example for cytoplasmic inheritance, we can say mitochondrial inheritance. Here too, a reciprocal cross was conducted, that is as in the case of Mirabilis gelata. Here if you are taking the cytoplasm, there are two different types of cytoplasm in the plants. One, plant with normal cytoplasm, that is designated as N, and plant with aberrant cytoplasm, which is designated as S because it produces sterility. Aberrant cytoplasm means defective cytoplasm. In the first cross, he crossed actually the person plant with a normal cytoplasm, which is a male. A male plant was taken with a normal cytoplasm, so it is fertile male as it contains norm. It is crossed with plant with aberrant cytoplasm, that is a female. Here the pollen grains are transferred to the female. So it is S plant. So F1 male sterile, it becomes sterile. If it is Mendelian reciprocal cross, we know in both the crosses we have received all same character or same trait. But here there are reciprocal differences. As in the case of Mirabilis gelata, here the character is transmitted from the female parent to the male. See that one, as the female parent is defective, having what is called aberrant cytoplasm, now the F1 male becomes sterile. This is one class. Now we are taking what is called the reciprocal class. Now we are taking the plant with the aberrant cytoplasm with reference to the male plant. It is not considered as a male plant because it produces pollen grains which are sterile, unable to fertilize, that is what is called the egg. And it is being crossed with what is called plant with a normal cytoplasm. It can be a female, so it is fertile in nature. Here crossing is not possible. The female plant produces fertile egg, whereas the male plant produces sterile, there is a pollen. So cross is not possible. That's why I'm writing no cross is possible. So anyway, here we can find reciprocal differences. In the case of Mendelian inheritance, there is no reciprocal difference. All the individuals in both the crosses are similar, having the same trait. But you can find here reciprocal differences. So it is because of the gene present in the mitochondria from the female parent that causes sterility if the gene is what we call the sterile gene. If the female parent has fertile gene, then the resultant male plant will be a fertile male. But here we are getting only the sterile male. This is due to because of the gene transfer from mother, that is to the male. So here we see that one here also the same one as in the case of Mirabilis gelaba, but the gene here is located in mitochondria. Students, let's have an explanation about the role of RF genes. So this is what is called fertility of restoring genes or restore fertility genes, restoring the fertility. To restore fertility in the case of abnormal cytoplasm or S cytoplasm. And we have to go for some of the facts. Now we know the nuclear genes I mentioned already, they follow the Mendelian inheritance. But in most of the plant species, you have cytoplasmic genetic sterility seen. Now this is because of both the nuclear genes as well as that is cytoplasmic genes. So the sterility is maintained both by the nuclear genes and the cytoplasmic genes of mitochondria, or even we can say chloroplast. Here we are talking about mitochondria. If you are taking the cytoplasm, there are two types of cytoplasm in plants. One, just in the case of maize I am talking about, one with the normal cytoplasm designated as N. Another one with S, that is cytoplasm, that is nothing but aberrant cytoplasm, as it is responsible for sterility, it is called S cytoplasm. In addition to these, actually the cytoplasm controlled by extranuclear genes, we have certain nuclear genes. They are called restore of fertility genes. Restore of fertility genes, it is normally a nuclear gene. It is simply called as RF genes. I mentioned earlier, it is a nuclear gene, not a cytoplasmic gene. It's a nuclear gene present in the chromosomes, but distinct from cytoplasmic male sterility genes in other plants. Distinct from cytoplasmic male sterility genes of other plants. Now, the RF genes do not have their own expression unless the presence of S cytoplasm. So under normal conditions, they cannot express. They can express themselves only in the presence of S cytoplasm in order to modify the S cytoplasm, making the cytoplasm fertile. Making the cytoplasm of the plant fertile. That's why it can express only in the presence of S cytoplasm. In the absence of S cytoplasm, its expression is not there. Okay, so the gene RF is required to restore the fertility of S cytoplasm. As I mentioned earlier, it is working only in the presence of S cytoplasm. In the absence of S cytoplasm, it doesn't have its expression. When S cytoplasm is present, it can express and modify the S cytoplasm so that the plants can produce fertile pollen. And the S cytoplasm is responsible in northern one sterility in males. So in order to make the male sterility plants into fertile plants, the S cytoplasm has been modified with the help of restore. Modified to restore what is called the fertility. Hence it is called restore of fertility genes. Restoring of fertility genes, or we can say restorer fertility genes. So this is one concept. I can represent the illustration with reference to all these facts, what's happening. Now, we have to go for the explanation related to the role of restore fertility genes. Now, RR genes. Now, a plant with a normal cytoplasm with RR genes, that is a recessive. This is normal cytoplasm with RR genes, and the plants with S cytoplasm, that is aberrant cytoplasm or sterility cytoplasm with the dominant R genes. And these two have the ability of producing plants with fertile pollens. That is normal cytoplasm with the recessive genes or S cytoplasm with the dominant genes produce plants with fertile pollen. Whereas a plant with aberrant cytoplasm or sterility cytoplasm with the recessive genes. So here both are defective. The recessive genes, there is no dominant restoring fertility genes. We have only the recessive genes. So when a plant is having a cytoplasm with what is called aberrant cytoplasm with recessive genes, they produce only the sterile males. So we need at least one capital R, that is to express its character so that to produce the fertile offsprings. Now we have two crosses. This is cross 1. Now in cross 1, we are taking normal fertile male. Now the genotype N, the recessive genes. So normal cytoplasm is there, even though recessive gene is present, now it is a fertile male. Then we have what is called aberrant cytoplasm, the female plant. Though S is there, the aberrant cytoplasm, because of the presence of RF genes, one dominant gene, now the female is normal. So a cross is affected by normal fertile male and female with the following genotypes. Now we are getting actually the plant with this genotype, that is a heterozygous, capital F, smaller f. Now this F1 male is fertile in nature. This is because of the presence of one dominant gene, namely this is capital R. So that's why F1 is fertile, the male is fertile due to the presence of restorer gene. The restorer gene is nothing but R gene. Here at least one R gene is there. Now I am making a cross too. Again normal fertile male with the genotype N, recessive homozygous. We are taking another one that is a female, the same genotype, heterozygous condition, S or F, then recessive alleles. Now here we are getting, so I am taking what's called, this is otherwise called the test cross. In a test cross, you know the probability 1:1. There is a possibility of getting a dominant parent or a recessive parent. So we are taking a heterozygous individual crossing pure homozygous. We will get either a dominant parent or the recessive parent. So in that case, we have received the dominant parent, that is when the male is fertile. In that case, this is also a test cross. There is no appearance of what is called the dominant gene. We have appearance of only both the recessive genes. As the restorer gene is absent, namely the dominant allele capital R, now the F1 individual is sterile in nature. So the gene is coming from only female. So the female is responsible for making the sterility. So individual with the S cytoplasm with the recessive genes always normally sterile males. So this is the individual with what we call, that is a recessive alleles with this what is called the cytoplasm. S cytoplasm or aberrant cytoplasm or sterility cytoplasm, not sterile, sterility cytoplasm, that results in the appearance of what is called sterile male. So for the production of fertile male, it needs what is called the restorer gene. That's why I say, now in the case of cytoplasmic inheritance or the mitochondrial inheritance, both nuclear genes as well as the cytoplasmic genes are taking place. Now N, that is what we call the mitochondria, the R gene, that is from the chromosome, and likewise N from chromosomes, and S also from the chromosomes, and these R genes normally we have, that is from the mitochondria. So anyway, we need the gene from a dominant gene from the female parent is responsible for making the male that is fertile. So this is what we call the interactions between the nuclear genes and the cytoplasmic genes to make the individual or to restore the fertility of the individual. Hence it is called restorer gene or restorer fertility genes or F gene. So atavism is a sudden reappearance of ancestral characters in one of the present generations. For example, presence of dense hairs on the surface of the body, the chest region in humans, or the presence of long canine teeth. It's an ancestral character. Or the presence of short stomach tail in the case of a newborn baby. These are examples for what we have in zoology or animals. Now what is atavism as given given in the book. So atavism is nothing but a biological modification, that is biological modifications in structures where reappearance of ancestral characters occur. And those traits which have been lost for a number of generations, which have been lost through what is called evolutionary changes in the previous generations. So those characters which have been lost through what we call evolutionary changes in the previous generations, and that is called atavism. Now in atavism, though the evolution traits have disappeared phenotypically, now the evolutionary traits have disappeared phenotypically, but they are not actually disappearing in the organism's DNA. So only the phenotypic character disappeared, but its presence is there in the DNA. It remains in the DNA. So the gene sequence is maintained in the DNA of an organism in the ancestors. Now, it is normally, you see that one, not disappeared I mentioned, but actually it remains in the DNA. So, the genes remain intact in the number of, actually intact in just the generations or in the genome of the organisms for many generations. So the gene remains intact and the gene sequence remains intact, that is in the genome of organisms for many generations. Suppose there is a fault in the unused genes. So in the unused gene, normally all of a sudden reappeared, express its character. What is the reason for that one? A fault. There is a fault in controlling the expression of unused gene. And this leads to the reappearance of ancestral characters. So the genes remain as such in the genome, but it is in inactive form. When there is a fault in controlling the expression of that unused gene, that leads to the reappearance of ancestral characters. That is a controlling gene that controls the expression of that unused gene. Sometimes what will happen, because of mutation, a fault occurs in controlling the expression of unused gene that results in the reappearance of ancestral characters. In plants, the best example, re-emergence of sexual reproduction. In the case of plant, Hieracium, Pilocellular. Now, this plant lost its capacity to reproduce sexually, and all of a sudden, in one generation, now re-emergence of sexual reproduction occurs in this plant. So it lost its sexual reproduction, but automatically in some generations and few generations, all of a sudden, the plant was able to reproduce. That is called the re-emergence of sexual reproduction. Re-emergence means the reappearance of the ancestral character. That is called actually atavism. I mentioned already in the case of animals. In the case of human beings, we have the long canine teeth and also dense hair on the surface of the chest, and also the presence of short stumpy tail. In the case of newborn babies, these are also examples for atavism. Simply we can say sudden reappearance of ancestral characters. This is called atavism, sudden reappearance of ancestral characters in one of the generations, in one of the present generations. Okay, so with this I concluded the lesson what we have the classical genetics, and which also completed what we have atavism, cytoplasmic inheritance, etc. This is the last part of the lesson, classical genetics. I would like to go to the next part in zoology, related to once again genetics, molecular basis of inheritance. That is also another important chapter. I will take it in the next